Hello guys. I was in the last video I was explaining the membrane attack complex. So once the complex is formed, the pores are formed on the cell membrane of the bacteria and other microorganisms, and so that the water, due to the osmotic barrier, the water enters into the bacteria and causes swelling and bursting of the cells. That is how the um, invading microorganisms are killed by the membrane attack complex or by the complement system. So this the classic pathway, the components of the membrane attack complex. Next, coming to the biological effects of complement, so which causes opsonization, that is increasing the phagocytosis by C3b and C1q, and chemotaxis, that is attracting of the cells C5a, C5a and 5, 6, 7 complex, so that attracts the neutrophils. C5a enhances the adhesiveness of the neutrophils to the endothelium, thereby acting as a chemotactic element. So next one, anaphylatoxin. Anaphylatoxin C3A, C4A, and C5A. So so the larger fragments of 3, that is C3B, C4B, C5B. These are all participate in the formation of the membrane attack complex. The smaller fragment like A, 3A, 4A, 5A. So these acts as anaphylatoxins, causes degranulation of the mast cells, bind directly to the smooth muscles of the bronchioles, leads to bronchospasm. So C3b is an opsonin that increases the phagocytic activity. Next, cytolysis, that is caused by the membrane attack complex, that is MAC, disrupting of the membrane and entry of water and electrolytes into the cell. Next, enhancement of the antibody production. So binding of C3b to its receptors on the surface of the microorganisms activates the B cells, thereby ending in production of the antibody. So next, the how the complement system is regulated, that is regulation of the complement system. So it is given here and it is also given afterwards. So complement system should be strictly regulated, otherwise it will damage and causes damage the self tissues and causes the autoimmune diseases. So the complement systems are inactivated through their spontaneous decay. So the first point is, so complement components are inactivated if they are not bound to the surface of the microorganisms. It will be inactivated. That means the free molecules of complements are inactivated immediately after few minutes. So example is given here. The newly activated C3b and C4b react with water with half-lives of approximately 60 microseconds. So after 60 microseconds, this will be inactivated. These proteins are therefore lost to the classical pathway. So no formation of the membrane attack complex, etc., unless they attach to a membrane in the immediate. So if the components of the complements are not attached to the surface of the microorganisms, then they will be inactivated. Okay, so activated molecules will be inactivated. Similarly, the the C3 convertase, the C3 convertase, that is C4b2A, is transiently active. Its 2A component is inactivated. 2A component is inactivated, means which loses the activity, which dissociates from the enzyme and loses its activity. Next, second one, the complement components are inactivated through their degradation by specific proteases. So specific proteases like C4b binding protein, which forms a complex with I and I, factor I, that proteolytically inactivates the C4b. So what is that? C4b binding protein forms a complex with factor I, and that complex inactivates the C4b. You have already seen IH complex, which degrades the C3b in the alternative pathway. Similarly, here also. Apparently, C4b binding protein and factor H acts as a cofactor that target the factor I for C4b and C3b. So from this point of view, C4b binding protein limits the activities of the classical pathway, that is C3 convertase and the C5 convertase, whereas the factor H does so for proteases containing C3b in both the classical and the alternative pathways. So what is it? So here, what is the inference? So complement components are inactivated through their degradation by specific proteases. So specific proteases here includes the factor I. So next, third one, coming to the third point of regulation. So the complement components are inactivated through their association with the specific binding proteins. Association with the specific binding proteins. So example here given is a C1 inhibitor. C1 inhibitor which tightly binds the C1R and C1S. C1 inhibitor tightly binds C1R and C1S to form a complex that dissociates from, okay, and inactivates the recognition unit. Recognition unit is C1. So C1 is inactivated by C1R and C1S complex. So similarly, one more protein that is known as S protein. So S protein attaches to MAC. S protein attaches to MAC, that is membrane attack complex, assembling in the plasma so as to prevent their later attachment to the cell membranes. That means S protein prevents the attachment of the MAC to the membrane. So such attachment is consequently limited to the site of complement activation. So this is how the complement system is regulated. This is also very, very important. So regulation of the complement system and the biological activities of the complement system is important. So the simplification here, it is given how it is regulated. So even inhibitor, factor H, piperidine, factor I, how these are all regulate the complement system is given here. Thus you can easily understand. So next, clinical aspects of the complement. So deficiency of the complements. What happens when the complements are deficient? When the complements are not present, what happens? So there are so many diseases which are caused due to the deficiency of the complement. So C5 and C8 deficiency, the first one, deficiency of C5, C8 and the menon binding lectin, that is lectin pathway, menon binding lectin, MBL. So that predisposed to severe Neisseria bacteremia, okay. And similarly, deficiency of C3. If C3 component is not present, severe recurrent pyogenic sinus and respiratory tract infections. Similarly, deficiency of C1 esterase inhibitor, which leads to increased capillary permeability and edema, deficiency of DAF, which causes complement-mediated hemolysis, transfusion mismatches, autoimmune diseases, and severe liver diseases. These are some of the clinical aspects when the complements are deficient. Okay, so factor I, etc. So this is about our complement system. So important things are, what are complements, the C1, the structure of C1 and all, and the classical pathway, alternative pathway, classical pathway and its regulation, alternative pathway and its regulation, and what else is the lectin pathway. So all these pathways converge to form the membrane attack complex. What is that? Classical pathway, alternative pathway, and the lectin pathway. All this converge in forming the membrane attack complex that leads to the formation of pores and the microorganisms, thereby entering the water inside the bacteria and it bursts out. So that is how the killing of the bacteria takes place. Okay, so the classical pathway requires the antibodies, so that takes place only during the acquired immunity, and the alternative pathway and the lectin pathway does not require antibodies, so they are active in during innate or for innate immunity. So they will be active for the innate immunity. And what is membrane attack complex, how it is regulated, what happens, the deficiency of the complements. So what are the clinical aspects, what are the biological effects of the complements. So this is about your complement system. So you go through once. So all I think I've given the some questions also I've sent. So you go through once. If you have any doubts, you come back to me. Okay. So you can call back or you can message, something like that. Okay. Okay. I'm finishing with this, the complement system, and I'm sending both two. That means 3 hours of this. Thank you.